Maybe let's get started with the agenda and then people can uh, come in as the meeting unfolds. Good morning, good afternoon, everyone. As usual, this meeting is going to be recorded, and we intend to share this on YouTube. So please reach out if you have any concern with the content before we release it on the platform. Okay. So our meeting agenda today is something that we discussed last week. So first, we will. I propose we just have a quick follow-up on the review with physiological review. I'm happy to answer any questions that this group has. And then I'm planning to present plans to lead a discussion on imaging data. And I'm hoping that we finish with a discussion on how to best classify cells in the imaging data and maybe also in the neuropixel data. Are there any questions on this agenda? Things we should bring up? Okay. So quick follow-up on the review. So I have sent emails through all co-authors of the physiological review at this point, and so we've exchanged on emails. The status is our review has been looked over by the editorial board of the physiological review, and they ask us to add more senior corresponding authors. And so as a group, we agreed to add Henry Kennedy, who is the most senior co-authors of the piece, and he agreed to do so. And so I am hoping that we start discussing with the board of the journal with Henry, with potentially the intent of trying to add a few more contributing orders, corresponding orders, um, especially given that the review is so large that it might be helpful to have more people expertise distributed across the piece. Are there any questions on this in this group? A subset of you have participated in a review. No particular concerns or okay. And the other aspects is I'm trying to aggregate a contribution table for the piece so that we clearly describe what everybody did. I think because so far it was just an alphabetical list, and I don't think we did a good job describing that. And so I'm hoping that we can improve on that in the next version. I've shared a link in a Google Sheet, and so feel free to fill in your contribution in this table. Are there any more questions on the review, or we should move on to the imaging presentation? Okay. So I have a separate set of slides. Okay, can you see my screen?
Yep.
Okay, so I have a few introduction slides for the content, just to support discussion, and I'm hoping that we mostly have a discussion. These slides is just a screenshot from our review, and what we highlighted in our shared review is that they are likely a distributed set of mechanisms that are implementing predictions, and that's potentially why it's so challenging to create concerted insight across all of those papers, because many of these are touching one or two of those mechanisms, but they're not trying to compare across. partly because of the way they record the data, and also because of the experimental design of each study. And our intent is to, as much as we can, with the resources that we have, cover as much of a larger range as we can possibly. And so the intent of our experimental design is to have multiple sensory prediction context, from sensory motor to purely visual context, with short-term and long-term predictions context in the past, and record all of those contexts with different technologies. So here we are highlighting 2-photon and NeuroPixels, and so these are two cohorts that we are planning to record from in all of the 4 different contexts. So sensory motor, standard oddball, sequential, and temporal jitter. And we are planning to do these both for the NeuroPixels and the 2-photon. And now we are talking with the SLAP2 team to do the same with SLAP2 data. And the challenge here is how we can do best use of all these technologies. And so the current experimental design has all of those oddball blocks at the onset. So you see there's a control block, followed with an oddball block, followed with another control block, so 1.1 and 1.2, with the intent of looking at drift and other similar phenomena, and eventually had multiple control blocks in the end that link each recording day. At the very end of the session, we have a receptor field mapping, which I see as a way to characterize the neuron properties outside of all of those contexts, so that we have a good baseline and also link to all of the previous literature on visual responses in those areas. So that's our current design. And our technology are complementary because NeuroPixels on one end has a high temporal resolution, but low spatial resolution. You can record a few neurons in one area, and in some cases, we don't know very well how they look at it with respect to one another. We can infer connection, but the advantage of having 2-photon is we have the complementary resolution. So low temporal resolution, but high spatial resolution with calcium imaging. So these are very much complementary, so that we can cover not only all of the cells in one area with imaging, but also their inputs coming from a distributed network with NeuroPixel probes distributed, as you see on this image on the top or the left. And the added bonus of having SLAP is that we can focus on a single cell, and with good imaging, we can see a majority or a large portion of the inputs, and so that provide both high temporal and high spatial resolution at a low scale. So all of those techniques, in my mind, are highly complementary, and so we should think carefully about how we design the experiment to leverage each of their strengths. So that's our current intent, and our initial plan for imaging was to have. So this is the table that is on the review for the experimental design, and so you see we have imaging cohort. So initially we were thinking of using a pan-excitatory and a pan-inhibitory GCAMP line to record as many as we can inhibitory neurons and excitatory neurons across V1 and LM. So we wanted to record densely in that area so that we can see how all of those inputs coming from distributed network recorded with NeuroPixels are integrated at that level. So from those plans, we started to discuss internally at the Institute what is the best way to leverage what we have. And we thought that the best way to do this was to leverage a recent pan-neuronal line that we have well established here that essentially express GCAMP8 across both excitatory and inhibitory neurons. So this is powerful because then now we're recording in the same animal both the excitatory and the inhibitory network densely. And so That's our current, we think, our best a shot at complementing the NeuroPixel recordings. Now, the challenge here is how do we know which one is which? Because we have both cells mixed together. And so there are several approach to do this. And the intent of the next few slides is to start the discussion. These slides is a suggestion from Saskia de Vries at the Allen Institute, who she highlighted a recent study from Daniel Millman that is in eLife, where they looked at functional response differences across those cell types. And so what you see here is analysis of the calcium data. This was recorded on the calcium imaging pipeline at the Allen Institute. And you see here, what's striking is that the VIP population, which is a subset of inhibitory cells, very much respond when the contrast is very low, the contrast of drifting gratings, and particularly to a given directions of drifting gratings that is going forward to backward. So it's probably related to sensory motor coupling in some ways in this particular case. So the VIP population has this signature, whereas the SST population, in contrast, tends to respond to a higher contrast. The excitatory population do not follow that patterns and have a more distributed direction tuning and a less striking change in response across contrast. So that's one way to do this. Another way that I've started to think about, given the previous discussion, is linking with some of the electron microscopy data that has been collected here in partnership with, at the time, Baylor. And so in this study that came out this year, they use a set of visual stimuli to characterize the functional properties of excitatory cells. So this, they did not, as far as I know, they did not record inhibitory cells in Booth's experiment, but they covered all of the excitatory cells that they could using a pan-neuronal line, as we are suggesting to do. And they, interestingly, they use a stimulus called TRPE that is extremely similar to the one we reviewed last week. Perhaps lower contrast. And so they have actually characterized the response of they have recorded the response of all of the excitatory cells to this orientation tuning stimulus. So I think that could be a potentially useful data set to linking with EM anatomy if we are to reuse something like TRPE. Or zebra, like we talked about last week. So that's one way. Right? A third way to do this. So this is what I'm thinking now. I think I had I had a third slide. Another idea is to use transcriptomic. So that's, we'll say, is the gold standard today to classify. And so the idea here now is to record the cells with calcium imaging, and then to take the brain at the end of the experiment and perform spatial transcriptomic to, with a couple rounds of labeling, identify the expression of those cells. I've had several discussions at the Institute whether we could do this because I felt this was the most compelling way, but it's very demanding to our resources, and currently OpenScope lacks sufficient resources to do that. And so that's currently where we are on this option. But I'm curious to hear everybody's thought on how we could do this. Okay, so these are the three options. Are there other ideas that you have, or that we should consider, or what's your opinion?
Jerome, the first method, how would you distinguish PV and SST neurons? Because right, that
So
would be require another kind of manipulation.
So PV have I think less sharper tuning than SSD. I think that's right.
That could be a marker, yeah. I don't know if you know, what's the actual discriminability, but because that's the question. In order to classify, these are averages, right? So
Yeah,
it would be interesting to re-examine this particular data set and then actually see what your cohens D, or in other words, if you would have some kind of area under the curve, can you actually reliably decode whether it's a VIP and SSD? In terms of orientation tuning strength, that tends to be measured with a lot of heterogeneity. So I'm not entirely sure whether one could use it. And surround size tuning could be another option, but I think that's also not that discriminative between SSD and performance endurance. So
that's right. I think they touch a little bit on PV in this study, and when I check, the VIP behavior here is strikingly homogeneous across the VIP cells that were labeled. I think there were maybe like a 10% that were outliers. I think these are selected for responsiveness before what you see. And so I feel like I agree with you that it's not going to be the cleanest that you could do, but it's one proxy. And I think I agree with you, but the PV is the most challenging probably of all. Although with NeuroPixels, we probably have the best classification of PV cells because they're fast spiking, right? So you might be
yeah. Well, you still have the subset of narrow waveform SSD neurons, but about 30%.
yeah.
You can classify them based on some things like the temporal phase, etc. Of firing. That actually is something we have used. Yeah. I mean, like flicker could also be interesting because the SSD neurons tend to be much less responsive to high frequency flicker, whereas the pervalbumin cells can follow that. Or they will have more elevated fine rates because of it.
I see. So you would had a short flicker block.
You could have a short flicker block because the SST cells are, yeah, they tend to act as low pass filters. I don't know whether for the imaging that will pan out. You would have to validate that somehow. But for the EFIS, that would definitely be a marker that you could use.
It's a good suggestion.
Yeah, because we have UpToTact SST PV, and they have very different responses to flicker.
In NeuroPixel, we'll have opto tagging, so we're planning to do that.
Okay, you do have it. Yeah.
It's just
Yeah.
Yeah.
You still have sparse expression, right? So you might have, you might benefit from having it still.
Any other thoughts on this challenge?
Yeah, so I think there's a lot to discuss. I mean, one question is what even is the ground truth? So, I mean, Martin just said, for example, what if a neuron doesn't express, something enough? You also have the opposite problem that isn't talked about in optogenetics a lot. If it does express a lot, the neuron might be dead by the time that you kill the neuron with your construct. You wouldn't even know by the time that you record. And so as you do these clustering diagrams, if you go by these heuristics and functionally, you start not just looking at overlap, but you just find out that when we say this is a VIP neuron or whatever neuron, it's like, you mean by the transcriptome or the proteome, or like what even becomes because these are gradients ultimately, it becomes difficult to chase what we want to chase for. So for that all that I'm saying here is I find the other two techniques more compelling. So if we use EM, if that's an option, there's a clear ground truth there, a physical ground truth that's lesser gradient. And obviously, if we could do the transcriptome, that would be the ultimate answer to all of this. So Jerome, you can tell us more about what might be needed here with these resources, or what would be feasible, or what would speak against using these instead of just a heuristic.
So I had many discussion about the possibility of doing transcriptomic at the Allen. So we have ongoing effort to establish this technology. The challenge is, I think the platform is able to record data, but it's all of the staff needed to monitor that process, do the registration and the analysis, and as you might guess, it's not a trivial, straightforward process. But you need people to manage that, and currently I do not have resources. And so that's part of the challenge.
And so that's true for both the EM and the transcriptomics. So both of these are
what I'm proposing with the EM is that there is an existing release data that has recorded pyramidal cells with a few stimuli that characterize the functional classes, and I feel like it will be interesting to present the same stimulus that were presented during that study to allow linkage.
I see. So we repeat some of the stuff that they did with Micron so that we can then leverage the fact that they did all the hard work about the EM reconstruction.
That's right. But they do not have inhibitory neurons recorded in this study. So it will be useful for pyramidal cells classes.
And so what speaks against combining that with what you're showing right here? So we could just basically improve on the functional heuristic maybe then?
So this is what I was, this is visualized essentially. So one thing we could do is make variants of the zebra trippy block with high and low contrast. Assuming, I mean, we haven't shown that, but the low contrast version is actually recruiting those different classes of inhibitory cells much better. It's possible. I mean, I would say it's possible that the high response in low contrast of the AP cells could be due to the shuffling. Like, so if you insert a low contrast variant among high contrast variants, it's possible that the network is like out of context and it suddenly detect that and just flash in the AP cells. But so that's one caveat. But assuming that this works, then I feel like these will be the most efficient way to map everyone.
So is the trippy already lower contrast? Is the trippy is on the right here?
No, trippy is in between. In addition, I also reach out to some of the people at Micron because they're here, and Dmitry Yatsenko, who was the lead in this stimulus design for the Micron, I'm just waiting on his confirmation, but he in principle agreed to present next week at this hour, and I just need to confirm.
And your suggestion is at the bottom that we replace the RF mapping with trippy instead of zebra, but we'll have one high contrast trippy, and that's more like zebra, except it's blurred. And then we have repeated with low contrast.
Something in between, actually something in between zebra and trippy, but essentially take the idea from both studies. I don't know what's the best way to do this, but it's just a suggestion.
I mean, it sounds a little bit that if we want to leverage microns in the way you suggested, we should exactly repeat the stimulus also,
That's
right?
right. That's right. So you see at the bottom right, this is their version is, I think, 80% contrast. So we could do that. Have like sort of multiple values.
I mean, one thing we get with the Gaussian filtering that they have, it's basically Zebra with Gaussian filtering, right? So it's just more Gabor-like instead of a step function or boxcar function. And so one thing you get with that for free is already that as you move from the darkest to the brightest parts of your image, you hit all contrast levels, right? So you basically, when we say 80% contrast, 100% contrast is kind of for the entire image. But locally, from a receptive field perspective, you get hit with all kinds of contrast levels as that sweeps across your region of the visual field.
When you do this on the left?
No, so if just what you showed, that trippy.
Yeah.
So you see how you have regions that are completely black, regions that are completely white, and then you have gray in between. And that's because as you low pass filter the transitions from black to white, you get all shades of gray. But that can be happening very, very quickly. But it means that you cover all contrast levels. And as that moves around, it moves across your receptor. So if you have just one receptive field, you get to see all of this, right? So in a way, when we're talking about the contrast, we just mean for the entire stimulus or on average or the median. But like from the perspective of a receptive field or V1 neuron, the trippy stimulus as shown here is not limited to all or none contrast.
Yeah, the receptive field in mouse V1 is they're pretty big, and then also they differ from cell type to cell type. So like SSTs have different sensitivities to large versus small stimuli, and then PVs have the opposite sensitivities. So it's true. It just it's trickier.
But Jordan, would you think that these differences will be I don't want to say sufficient, but will be useful to build models on top? Like, I mean, are we going to be able to assign?
Yeah. I don't know, but what I feel is that, whether or not you know if cell X is SST layer 4 or not, might not be completely necessary if you know that cell X has these other interesting properties. So yeah, I don't know. I guess it comes down to like, what classification criterion are we thinking of as ground truth? I mean, Alex was saying this as well. It's like, if we're interested in linking it back to contrast sensitivity and locomotion related gain, which we know to differ pretty sharply between VIPs and SSTs, then that's pretty neat, I think. And I think like understanding which of those is sensitive to which types of prediction errors is also useful. So I don't know. I think it starts to get to be slippery ground when we ask the question, like, what are the cell types? Like, how do we carve things apart, and which of those different criteria do we think of as the ground truth in it, in the truly useful categories? And I think it just comes back to, well, it would be really wonderful if we had spatial omics. But then, yeah, I appreciate the fact that that's going to be extremely resource intensive, time and money. I do like this that's on the screen right now, because EM. So how much more difficult would it be to kind of image and then co-register to EM as opposed to spatial omics?
You mean the mouse we record from?
Yeah.
Even more expensive.
Even more expensive. Okay.
Because the EM is yeah, you have to slice the brain with nanometer resolution. I mean, we need to get a grant to do that.
Yeah. Yeah. Right. I don't think it's I don't think it's an awful idea. Let's put it another way. I think it's potentially useful to do this kind of contrast mapping that you're proposing and relating it to these other properties. For sure. I don't think it's going to be the end all be all of telling us cell types that are doing what and where. But I don't know. Who's to say which one is the most important? I mean, I think if you have like one subset of inhibitory neurons that suppress the prediction errors and one that's activated to prediction errors, maybe that's maybe that in and of itself is a useful category. I don't know.
In my mind, I'll tell me if I'm wrong, but in my mind, I would rather record all of them than a subset at one point. Like I would rather record all of them at the same time than record a subset of inhibitory neurons across multiple mice, because I feel like the variability across mice and the potential of having the concerted dynamics of all of the cells is more useful than classifying them using Cree lines.
I like that. I agree with that. I think, yeah.
I think this is to one point that Sarah alluded to is like, yeah. So Sarah said, I'm confused about the requirement to map contrast sensitivity as we are not testing anything related to contrast sensitivity in the proposed paradigm. Isn't the point to understand whether distinct types of cell respond differently to deviance? That's right. I think the point about contrast is that the classification of VIP cells is so strong with contrast that we'll have a really, I think we might have a clean answer by linking to other studies. And people have shown that VIP cells are important in those paradigms. And so knowing which are potentially VIPs is helpful. I think that's my my take, but I mean, including, yeah.
Can you go back then to the slide of your proposed experiment that you showed at the end? This one. So for the VIP detection, when you say high contrast trippy and low contrast trippy, are there two levels that would be especially beneficial to detect VIP by contrasting these?
Sorry, can you repeat the question?
So my understanding is that you say, let's do 5-minute high contrast trippy and 5-minute low contrast trippy, and that will allow us to detect VIP neurons in the 2-photon imaging. Is that correct?
That's right.
And so what contrast levels are especially good in that case?
These are on this figure. You see, when you get to 10 to 5% contrast, the VIP activation explodes.
So we're talking really low contrast then.
That's right. And I think it's because the system is in a different state. Like you have to integrate information very differently when contrast is low. And so arguably the VIP cells are probably poking at the system to change its state. Because of their,
one point. So not with the natural images. This is with the grading. So with natural images, even at high contrasts, as long as they're novel, we get responses.
That's right. I mean, this is another.
Yeah, that's the
One could argue VIP cells are not. The contrast behavior is just a nippy phenomenon of why it happens, and I think if you look at across the literature, clearly VIP are involved when the prediction state is different, and potentially when it's just sort of challenging either the bottom up or the top down inputs, and so it switched the inputs, and so when you switch from novel to a familiar, they also sort of engage. And so I think like there's different cases where it happens.
So my understanding, we had another discussion about whether we use Trippy or Zebra, but my understanding is, I mean, they're so similar, right? So the only difference is basically the spatial frequency content, whether you low pass filter the edges or you don't. And the real interesting thing about Zebra, though, is that it's two things. It's the stimulus and then an analysis package. And the analysis package, by treating every neuron as a Gaussian, they were able to reconstruct all of these neuronal properties, receptive field, spatial field. So I think that it probably still works with Trippy, though, right? So probably that we could still use and leverage.
I assume so. I assume that's the case. And
so that it would be no
we're getting into a state where,
yeah,
we're getting into a state where I mean, like this. I think it's a good idea that multiple labs came up with, and yeah.
So then there might be no real downside to using Trippy, but if we use Trippy, we gain the fact that we can relate it to microns.
That's what I'm trying, and then it comes out ultra distorted like I'm watching a movie.
Sorry, Eli, I muted. You had like a microphone. Okay. I think so. I think that's what I would argue, Alex. But that's why I reach out to Dimitri and get his perspective over the weekend. And one thing that we could do is, well, the NeuroPixel pilot is, our planning for the NeuroPixel pilot. I have it here. Right. So we are 2 weeks away from starting recording, and so I think we could design a few stimulus block, have a mouse, watch it, and then do analysis on it to make sure that it's what we want. I'm happy to work towards this. That would allow us to answer some of this unknown. So I don't know, maybe just play Trippy and Zebra at the same time.
That'd be very interesting. And so you will get the code for Trippy also in time that you can implement that?
I'm assuming both of these are studies that have been released. So I think Zebra, we have the code. Trippy is published. So I assume they will share it.
The code is not published. So they have a GitHub archive, but they didn't publish the code for it. They give enough description that I could try to recreate it, but their code is not published.
Okay. I mean, we could ask them if they're fine with sharing. And
yeah.
Okay, so Farzaneh, do you want to comment on your post?
Sure, yeah. So I was just saying that, so we had this preprint recently, as some of you might know. So it actually is not entirely true. So we did see a large fraction, as I'm showing, like for sure, most of the VIPs are inhibited with the high contrast, but we saw something like 25% of them also activated. So what we say about VIP being inhibited at high contrast, that's just if you just average everything. But we did this very based on we did this temporal receptive field model and use that for clustering. And I'm just saying that there is a large fraction of VIP that is not. So I don't think that idea of VIP encoding anything that is either surprising or not quite predictable is valid for the whole population.
And Farzaneh, did you present multiple contrast in your study?
No. So this is just full contrast.
But how did you know? How do you know?
So we know that a good fraction, like 25%, 30%, do respond to the high contrast. So not all.
Oh, I see.
Yeah, but the question for us was not varying contrast. I just know that with the full contrast, 100%, we do have a large population of VIP that is active by it. Most are
Yeah.
inhibited, but a substantial fraction are activated as well across both PPC and visual cortex.
I see.
So I just think VIP does more than the one thing that we try to summarize it.
Yeah, I mean, it's going to depend on which
Functional cluster we're talking about.
But I was reading the microns. They have a recent paper. I really recommend it from the group about inhibitory subclasses as shown by connectomics, and they discover a subclass. I think they claim VIP cells that do not have the same projection output. So it's interesting.
Right. Yeah, it would be really cool to know like the two categories of stimulus activated and inhibited have what it means in terms of their input output.
Okay.
Yeah.
What does the group think which our next step should be for, like, given that we have a pilot coming and then we could integrate some of the stimulus block to pilot? I'm just curious what people think. I think Alex, you alluded to have several contrast. What do you all think? Should we try to have Trippy and Zebra?
I mean, I have a long background in studying early visual system, and certainly from that perspective, having different contrast levels would be extremely helpful. I mean, it's as basic as all the other types of tuning that we look at, and given that these neurons are so non-linear in their contrast response function, I think that yes, this would be an added piece of information that would certainly be helpful. Now, it sounds like we would be going very extreme, right? So very, very low contrast, 5% something, or then maybe really high, 85% or more. So yeah, that's interesting. I think that's interesting. And so I think for comparing Trippy and Zebra, the two things I think that have been discussed that I agree with is probably Trippy will give us greater responsiveness. And whether we want to sacrifice that for getting Zebra, sorry, yeah, Zebra will give us better responsiveness, but Trippy then has these added benefits of that it allows us to work with microns. And again, hardcore old school visual scientists would be more happy with a low pass filtered stimulus, given the receptive field properties of these early neurons. But the project is not just about V1, right? So that's why I'm careful to say all of that.
It sounds like the best would be to have free contrast. The low, the Trippy contrast, and the really high contrast of Zebra. Because this way we cover, and we could test this in the pilot.
Yeah.
So essentially make free variants of Trippy.
I like that. This is going to be a naive question, but is there a way we can preserve the tissue such that in the future we could then apply for a grant to do EM reconstruction, at least in some of the best data sets that we have?
I certainly can look into that.
Yeah, I guess because it's just that I think morphology, especially given the heterogeneity of yes, Sarah doesn't think EM is reasonable. Yeah, right. I'm not saying do it now. I'm just wondering if there's a way we could leave the door open in the future, because I think that would really give us great ground truth in terms of morphology of the cells, where which I think is just as ground truthy as transcriptomics in some cases. So anyways, I don't know how well it can be preserved for that. But I just wanted to float that possibility.
So transcriptomic, I've talked about this. For doing transcriptomic postdoc, the challenge is that, yeah, after a couple months, what is the challenge? Yeah, you need to maintain some of the staining, and so after a couple of months, that cannot be done anymore. But for EM, I mean, I don't know if you perfuse and freeze it. I will have to talk to some of the team to know what's possible.
Okay, yeah. I mean, again, it's not like every single experiment, but if we had like one mouse where we had a great variety of dynamics and a great number of cells, it could just be a nice.
Yes.
Yeah.
I mean, I agree with you. I think, John, I think it's unfortunate that all of this multimodal studies have focused on randomized stimuli. Because I feel like there's if there were some kind of prediction context, I think that would have because that's what the cortex is doing. And so I feel like it's unfortunate. I think that many people call for the need for prediction kind of experiment plus those multimodal mapping. Okay, so Sarah said that it can be stored, but not trivial, and she doesn't think one mouse will do the trick. So I can look into how long these can be stored. Would any of you have interest in taking some of the samples, or I'm just curious?
I might. I want to look into that. I have colleagues that I think we have an EM service on campus. I have to look into that.
Okay. Okay, so Sarah said the questions are all interesting, but I think we lose focus.
Sorry. I'm glad Sarah can't talk because otherwise I would have been put in my place and shamed out loud instead of just in the chat.
So it sounds like there is agreement that recording as many cells as possible is the best path. And then we can tweak the stimulus to better map their classes, but it's not a killer. Is that right? Alex, would you be able to help with testing some of those stimuli on your end, or?
I can. All I'm doing is run it on my laptop, so I'd be happy to do that. Yeah.
Because I think what I need is some way to pre-generate the video and then deploy it on the rig. And then but I would need this within the next week or so. I don't know what's your bandwidth, but if we do this efficiently, I think I could deploy it on the NeuroPixel experiment and we can do some comparison.
So what would really help, Jerome, is if you can take a high FPS video, because the graphics card and monitor that you have, that may cause different actual physical frame rates. And so if we want to go after that, if you can record at 120 FPS, for example, then I could analyze that frame by frame.
So I think the way I would do this is I would replay a video at 60 frames per second. Oh, you want to, what do you mean by
if you can record the video at 120 frames per second, then we can analyze whether there are any frame drops up to 60 frames per second. So
I see.
The Nyquist frequency is twice. The Nyquist frequency is twice the sampling frequency.
Oh, I see. Well, yeah. Just on this, I've done a lot of bonsai improvement in the last 2 weeks about yeah. So I've actually improved there was a bug in our code that was delaying the frame display, and so I think I fixed that. And so I actually have I can share it with you. I have data about how well the graphic card was following the command, and now it's at 60 hertz. So it's a complicated. We used to have a texture that was converting the screen to blue for SLAP 2, and that was delaying the refresh. That's why I removed that, and now it's very fast. So I'm not worrying about playing at 60 hertz. What we could do is if you're able to generate a 60 hertz pre-recorded movie with Trippy or over variants, I can play it during the experiment, and we record the frame number.
Got it. So that would be like a more for MP4 file, 60
That's
frames
right.
per second.
Yeah, and then maybe we should try to have free contrast.
I give that a go. Yep.
Thank you. Well, thank you for the discussion. I feel like we should continue on the forum as usual to make sure that we are testing this properly. It sounds like we have a plan. Are there any? So I'm gonna move forward with trying to get Dimitri to present. I think they have an incoming presentation at SFN, so hopefully he can. He has this content ready to present to us. I'm just not sure, but I think it would be nice if he can share what they have learned from Trippy in their own data. And so hopefully that happens next week. But other than that, are there any progress on SLAP 2 that we should discuss, or I wasn't sure if some of you have looked at the newer data.
So I wrote code that does a basic quick plot of the responses. And so for the fixed files, it seems good now. And so I haven't seen if anybody else has taken up the torch. I'm too busy at the moment.
Okay.
But just to double check, Jerome, do we now also have, in addition to these inputs to each of these synapses, do we have the glutamate spiking of the neurons as well, the output?
Yes, so we have done
Not
some
calcium. Sorry, calcium.
We have done one recording, a couple recording with it. I think it's packaged in the second channel. I'm going to ask Carter to follow up on that, but it's just going to bleach very quickly.
It's still worth looking at. So yeah, if we have that package, then I'd like to write some code quick to get that.
Yeah, I don't know if Andrew, you want to comment on this. I don't know. He's not online. Yeah. Okay. I'm going to send a couple emails to the folks that did analysis to see whether they're in progress. And Sarah said, "Oh, okay. I have students starting to join. Will it be possible for Carter to give an update to people joining sometimes?" Okay. Sure. I mean, what else should we discuss? Anybody has inputs that they want to share?
I just wanted to ask for the analysis the most recent three SLAP 2 data sets, they are all okay, right?
That's right.
Analysis. Okay, perfect.
So these have the design that you suggested we do. So the multiple blocks. I'm assuming you're going to be able to test some of your ideas.
Yeah, perfect. Yeah, we're going to pick this up next week or next 2 weeks.
Okay, thank you. Fantastic. Okay, so hopefully next week we have a presentation from Dimitri, and then we can go from there. Oh, Andrew, do you have a comment?
Yeah, sorry, it took me a second to plug in my webcam. just to be clear about the SLAP2 data, for the sessions that do include calcium imaging data, that will be included in channel 2. And the current best lifetime for the indicator that we have in those sessions is approximately 20 minutes. So the first third of the session should be good to get calcium data out of.
So just to be clear, that's the soma or also the dendrites?
It is somatic calcium. There is a calcium indicator in the dendrites, but I haven't looked at the signal for dendrites. But for recordings where we did record calcium, we recorded the soma in addition to the dendrites.
Okay, thanks. Yeah.
On SLAP2, we are in discussion internally to make a data collection plan until March. And the intent is to replicate one of the cohort in that we discuss for SLAP2 as well. I want us to align internally before we share it with this group, but I think that's the intent. Okay. Well, let's regroup next week. Thanks, everyone.
Thanks, bye.